Hello everyone, Zio on this side. So today hand we are dealing with the beta oxidation of fatty acids. So let's get started. First of all, we have to deal with the definition of the beta oxidation. So basically, beta oxidation of fatty acid is the breakdown of fatty acid or the splitting of fatty acid into acetyl coenzyme A and a fatty acid which is having two carbon atom less than the reacting fatty acid. Okay, deal with. For example, if the reaction is starting with the fatty acid having 18 carbon, so at the end of the day, it will be splitted up into a two carbon molecule that is called as acetyl coenzyme A and another that is called another fatty acid which is having 16 carbon molecules. Okay, deal with. Now, here. So the first step occurs in the mitochondria, or rather I'll be dealing with the beta oxidation of the fatty acid completely occurs in the mitochondria only. Okay, deal with. So here I'm starting with directly the first step, but still I'll be explaining that as well. The first step is called as the activation of fatty acid. So how come? Basically the fatty acids combine with coenzyme A with the help of the ATP to form fatty acyl coenzyme A. Here you can observe. Form fatty acyl coenzyme A. And the enzyme which is responsible for this reaction is called as acyl coenzyme A synthetase. Deal with. So this is our fatty acyl coenzyme A molecule. Okay? Deal with. Now, dealing with the second step that is called as a dehydrogenation first. So here the enzyme responsible is called as acyl coenzyme A dehydrogenase. The enzyme is called as acyl coenzyme A dehydrogenase, and here you will be observing that a new molecule is getting formed. So what happens in the dehydrogenation number 1? So basically it occurs in the alpha and the beta carbon of the fatty acyl coenzyme A catalyzed by the enzyme called as acyl coenzyme A dehydrogenase in presence of FAD. In presence of FAD. And the conversion of FAD will take place as FADH2 because the reaction in the reaction dehydrogenation is occurring. So ultimately whatever the hydrogens which are evolved from this reaction, they will be adsorbed in the FAD molecule forming the FADH2. Okay, deal with. Now if I will be dealing with the second one here, deal with. So here in this case we are having enol coenzyme A molecule. We will be having enoyl coenzyme A molecule, or rather this is called as unsaturated acyl coenzyme A. This is unsaturated acyl coenzyme A. You will be observing the double bonds here. You will be observing the double bonds here. So now whenever we are dealing with this reaction, basically this unsaturated acyl coenzyme A molecule is unstable. So again you have to add the dihydrogen monoxide, or rather we call as a water molecule into it. So the next step will be called as hydration, which means the addition of water molecule. The addition of water molecule. So basically in this step, the water molecule is added to the double bond of the alpha beta unsaturated acyl coenzyme A to form a new molecule. That molecule is called as hydroxyacyl coenzyme A. That molecule is called as hydroxyacyl coenzyme A. And the enzyme which is responsible in this is called as acyl coenzyme A hydratase. Acyl coenzyme A hydratase. Deal with the case. Okay. Now here. So now we are having hydroxyacyl coenzyme A molecule, and again we are dealing with the reaction called as a dehydrogenation number 2. This time the dehydrogenation number 2 will coming into scene because dehydrogenation number 1 has already happened. So in case of the dehydrogenation number 2, the hydroxyacyl coenzyme A is dehydrogenated to form ketoacyl coenzyme A. Here you will observing the ketoacyl coenzyme A here. Deal with. Correct? We are forming here the ketoacyl coenzyme A. This hydroxyacyl coenzyme A has given ketoacyl coenzyme A with the help of an enzyme called as hydroxyacyl coenzyme A dehydrogenase. Hydroxyacyl coenzyme A dehydrogenase. So again if the dehydrogenation is taking place, another molecule will be coming called as NAD plus. and this NAD plus will be getting converted into NADH plus H plus. Do remember one thing, that whatever the NAD and the FADH2, whatever the FADH2 we are having, and whatever the NADH plus H plus we are having, please do remember one thing, that these molecules are the one which will be yielding electrons in the electron transport chain, which we have already discussed. In the yesterday lecture, that this FADH2 and this NADH plus H plus will be yielding ATPs in the electron transport chain and the oxidative phosphorylation. So please deal with that case as well. Done? Okay. So now what we are having? We are having beta ketoacyl coenzyme A. So dealing with that case now. Here. So now, in this case of having beta ketoacyl coenzyme A, now our final step of the beta oxidation is taking place, called as cleavage. And the meaning of the cleavage is breakdown of one complex molecule into two different molecules. Okay, deal with. So here what we will be observing, what we will be having, that the last step of the fatty acid oxidation is catalyzed by the enzyme called as ketothiolase. Deal with. Okay. Now, a free coenzyme A is added. To the beta ketoacyl coenzyme A. So ultimately here now you will be having two coenzyme A. The primary coenzyme A from the ketoacyl, here you are getting the case. The primary is the coenzyme A here, and the secondary free coenzyme A has been added as well. So here now you are having two coenzyme A. So whenever this beta ketoacyl coenzyme A is getting splitted, it will be forming one molecule of acetyl coenzyme A and the one molecule of fatty acid. It will be forming one molecule of acetyl coenzyme A and one molecule of fatty acyl coenzyme A. So now in this case the acetyl coenzyme A will be having two carbon. Whereas the fatty acyl coenzyme A will be having two carbons less than the parent one. So deal with that case as well. Okay, 2 carbon less than the parent one. So whenever we will be running this beta oxidation of the fatty acid, do remember one case, that the fatty acyl coenzyme A will be re-entering in the next beta oxidation. And ultimately it will be releasing another acetyl coenzyme A and one more fatty acyl coenzyme A, which will be having 2 carbon less than the parent one. And this cycle will be repeated until the fatty acid is completely splitted up into acetyl coenzyme A. So basically, if the starting fatty acid contains even number of the carbon atom, for example, let's say if the starting one was containing 18 number carbon atom, 18. Okay, if the first one is having the even number of the carbon atom, the final cleavage result into two molecules of acetyl coenzyme A, because from this 18 you will be getting 16, 16 to 14, 14 to 12, 12 to 10, and so on. Like at the end of the day you will be having 4. This 4 will be split into 2 plus 2 acetyl coenzyme A. 2 plus 2 acetyl coenzyme A. Okay, deal with. Now if the reaction is starting from the odd number, so at the end of the day you will be having 5 carbon, correct? So this 5 carbon will be divided into 2 acetyl coenzyme A, which means one acetyl coenzyme A having 2 carbon and a 3 carbon molecule. And the name of this 3 carbon molecule will be called as propionyl coenzyme A. This will be called as propionyl coenzyme A. Okay, deal with. This is called as propionyl coenzyme A. Okay, deal with. So ultimately, hopefully you do know. If I'll be giving you if you will be asking a question that in a fatty acid chain there are 98 carbon molecules, how many acetyl coenzyme A will be formed from this, and those acetyl coenzyme A will be entering in the Krebs cycle? So like you just need to divide it by 2. And you'll be getting how much? You'll be getting 49 acetyl coenzyme A. Done? Okay. And if I'm asking, you are having 45 carbon chain in the fatty acid, what you will be doing? Just remove 3 from it. Just remove 3 from it, so it will be having 42, and this 42 divided by 2, you will be having 21. And this 3 will be called as propionyl coenzyme. This 3 will be forming the propionyl coenzyme. So hopefully you are getting the cases. This was all about the beta oxidation of the fatty acid and the formation of one molecule of FADH2, one molecule of FADH2, correct? One molecule of NADH plus H plus, correct? One molecule of NADH plus H plus and acetyl coenzyme depending on the length and the nature of the fatty acid. If it is of even number, all of the molecules will be divided by 2 and you will be getting the half number of the acetyl coenzyme, whatever the number of the carbons are present in the parent fatty acid. Let's say if the 20 carbons are there, you will be getting 10 acetyl coenzyme. And if the fatty acid is having like the odd number of the carbon atoms, you have to just subtract 3 from there, and that will be forming the propionyl coenzyme, and then whatever the remaining number is there, divided by 2. For example, if the 19 is there, subtract 3, you will be getting 16. 16 by 2 will be 8. So 8 molecules of acetyl coenzyme and 1 molecule of propionyl coenzyme. Done? Okay. So hopefully this lecture series has been beneficial for you guys. Thank you very much for your so much support. Please ask any kind of question if you do have. You can ask me any kind of the question like related to the topic itself. Done? Okay. Thank you.